The major function of the respiratory system is to supply the body with oxygen and dispose of carbon dioxide. At sea level, the air consists of approximately 21% oxygen, which has a partial pressure of 159 millimeters of mercury. Air is taken in through the nasal cavity and/or the mouth and travels down the pharynx, the larynx, and into the trachea. At the mid-thorax, the trachea divides into two main bronchi. The bronchi are repeatedly divided into smaller and smaller passages. The conducting zone finishes with the terminal bronchioles, which are less than half a millimeter in diameter. The respiratory zone begins with the respiratory bronchioles that open into the alveolar duct. Protruding from these ducts are scattered terminal clusters of alveoli called alveolar sacs. On the external surface of the alveoli are the pulmonary capillaries. The respiratory membrane consists of a single layer of squamous epithelial cells, a single layer of capillary endothelial cells, and a shared flimsy basement membrane. It is through the respiratory membrane that gas exchange occurs. In the alveoli of the lungs, the partial pressure of oxygen is 104 millimeters of mercury, while the blood's partial pressure of oxygen in the capillaries is 40 millimeters of mercury. Therefore, oxygen moves from the alveoli through the respiratory membrane and into the capillaries. Oxygen is poorly soluble in aqueous solutions. Therefore, only 1.5% of the oxygen is dissolved in plasma. The rest is bound to hemoglobin. Hemoglobin is contained within the red blood cells. This prevents the proteinaceous hemoglobin from making the blood too viscous and adversely affecting the osmotic pressure within the capillaries. One hemoglobin consists of 4 ring-like heme pigments and a globin protein. The globin protein consists of 4 polypeptide chains, 2 alpha and 2 beta chains, each binding a heme group. Within each heme group is an iron atom, which can bind reversibly with 1 molecule of oxygen. There are also hydrogen ions bound to the globin protein, which dissociate when oxygen binds. When the first oxygen molecule binds to the heme group, it changes the shape of the hemoglobin molecule. This change in shape allows the next oxygen molecule to bind more readily. The oxygen-loaded form of hemoglobin is called oxyhemoglobin. A single red blood cell contains around 250 million hemoglobin molecules, and therefore each of these cells can carry approximately 1 billion molecules of oxygen. These oxygen-laden red blood cells then travel to the tissues. The blood in capillaries arriving at the tissues has a partial pressure of oxygen of 100 millimeters of mercury, while the partial pressure of oxygen in the tissues is less than 40 millimeters of mercury. Therefore, oxygen dissociates from hemoglobin and diffuses into the tissues. As tissues metabolize glucose and use oxygen, they release carbon dioxide, increasing the partial pressure of carbon dioxide in the tissues to greater than 45 millimeters of mercury. The blood in the capillaries nearby has a partial pressure of carbon dioxide of 40 millimeters of mercury. Therefore, carbon dioxide diffuses from the tissues and into the blood. Carbon dioxide is approximately 20 times more soluble than oxygen, and therefore 7 to 10% of carbon dioxide is transported by the blood dissolved in plasma. Approximately 90% of carbon dioxide enters the red blood cell. Of that, 20% binds directly to hemoglobin, which is now called carbaminohemoglobin. The carbon dioxide is not competing with oxygen, since the carbon dioxide binds to the amino acids of the globin protein rather than to the heme molecules. The other 70% of carbon dioxide in the red blood cell is converted to bicarbonate ions. The carbon dioxide combines with water to form carbonic acid, and this reaction is catalyzed by an enzyme called carbonic anhydrase. This reaction occurs fastest in the red blood cells as they contain carbonic anhydrase, while plasma does not. Carbonic acid is unstable. It quickly dissociates into hydrogen ions and bicarbonate ions. The bicarbonate ions quickly move into the plasma. This means there is a rapid outrush of negatively charged ions from the red blood cells. To counteract this, there is an ion exchange process called the chloride shift. Chloride anions enter into the red blood cell via facilitated diffusion through the chloride bicarbonate exchanger, situated in the membrane of the red blood cell. The hydrogen ions generated by carbonic acid dissociation would decrease pH. However, the hydrogen ions bind to histidines within the globin protein, triggering the Bohr effect. The buffering effect of hemoglobin means there is little change in pH under resting conditions. The red blood cells then travel back to the lungs. Within the lungs, the partial pressure of carbon dioxide is 40 millimeters of mercury, while in the blood of the capillaries is 45 millimeters of mercury. Therefore, the reactions that occurred in the red blood cells near the tissues are reversed. Bicarbonate ions travel from the plasma into the red blood cell through the chloride bicarbonate exchanger. The negatively charged ions are again balanced via the chloride shift. Within the cytoplasm of the red blood cells, the bicarbonate ions join with hydrogen ions that were dissociated when oxygen bound to the heme group. Carbonic acid is formed. Carbonic anhydrase converts carbonic acid into carbon dioxide and water. At the same time, carbon dioxide is released from carbaminohemoglobin. All this carbon dioxide, including the 7 to 10% that was simply dissolved in plasma, diffuse across the respiratory membrane and into the alveoli. The carbon dioxide travels back up through the respiratory and conducting zones of the lungs and exits via the upper respiratory tract.